and oh, okay. Recording. So when
start the recording.
so when RNA folds, it coordinates magnesium, and that releases waters from magnesium. So that's just an example, like when you synthesize DNA or RNA or bind proteins to things, water is changing its chemical state like mad all over biology. And here, you know, hydrolysis of ATP, I mean, it's hydrolysis of ATP, right? So that means there's a water molecule involved. There's just water everywhere in chemistry. So if you think about biochemistry, on the left is the standard view of metabolites in biochemistry, and glutamate is generally listed as the, like in your cells, this is by concentration in a cell. Glutamate is the most highly concentrated metabolite in your cells. And then the other thing, see here is ATP is listed here. But really, those are just tiny compared to water. You know, the concentration of water in a cell is much higher. And then the number of water molecules that participate in biochemical reactions, it just exceeds everything by many orders of magnitude. And I think the number we came up with was something like, for an E. coli to replicate, every water molecule in that E. coli, well, on average, the average water molecule is a chemical participant in at least 3 different reactions. And that means a chemical participant. That doesn't mean just stabilizing a transition state or anything like that. So water is just this highly reactive medium. It's not just the medium. In fact, what we concluded after all this work is that the distinction between medium and chemical participant is not meaningful. That it's so blurred that you can't say, is water the medium or is water a chemical participant? It's like the medium is the chemistry or something like that. And you know, we think that, you know, if you want to understand the origin of life, you really need to understand the chemistry of water on the ancient Earth, and you have to think about the emergence of biopolymers, et cetera, in the context of water. So really, this is how we say it. You know, water is the medium, but water is the dominant chemical reagent. It's the dominant chemical metabolite of biology. And really, I mean, it's kind of funny, like David is going to teach biochemistry right now. He should really just ignore everything except water. Biochemistry, he's not going to, but biochemistry is really the chemistry of water. That's really what it is to a first approximation. That's not how we think about it. That's not how we teach it, but that's essentially what it is. Okay, so now I want to take you back to your Gen Chem, which you probably didn't like very much. And I want to talk to you about something called Le Chatelier's principle, because this is important in this whole water thing. And that's if you remember, actually, I've been talking to some geologists, and somehow Le Chatelier's principle is not necessarily part of geology. In fact, when I told them about it, they like asked me for a citation or something. I said, you know, it's just in every Gen Chem book, go look it up. But anyway, what it means is if you have a system at equilibrium and you add a reactant, it will push things to the right and you will get more product. So, and the other, yeah, so, or if you take away one of the reactants, the system will respond in the opposite direction, right? So this is, I don't know, maybe everybody knows this, but this is called Le Chatelier's principle. So now let's go back to water and think about it like this. We can imagine an equilibrium. Let's say we're synthesizing protein between 2 amino acids and a peptide. We just think about this in terms of not the net reaction, not the detailed reaction. So you take 2 amino acids, you link them together, and that produces water. What that means is if you have a system of high water activity, you're going to push this reaction to the left. When you add water to this system, you're going to push it to the left. Or another way to say this is that protein in in aqueous media is going to spontaneously hydrolyze. That's what this says. And it turns out that this applies to RNA and DNA. I have RNA here. So when you add water to RNA, you push it to the left and you hydrolyze it. When you add water to carbohydrate, you push it to the left and you hydrolyze it. So just the sort of, or let's talk about it going the forward direction. What it really says is that all biopolymers, all universal biopolymers, and a lot of other molecules in biology are made by condensation dehydration. That means you take two things, you put them together, and you get water out. Or you take the polymer, you put water in, and you break it up. So now let's think about Le Chatelier's principle. What this says is that in aqueous media, you are only going to have monomers. Thermodynamically, you cannot have a polymer in aqueous media because the equilibrium is going to go to the left. So then you say, well, obviously we have proteins and we have RNA and DNA and we have cellulose. So how does that work? These molecules are what are called kinetically trapped. They are not at thermodynamic equilibrium. The thermodynamic equilibrium is on the left, but they are kinetically trapped. And RNA, you would say, is the kinetic trap is not as deep as the other one. That's why RNA does hydrolyze pretty quickly. You can sort of see it go. You can almost watch it on your benchtop sometimes. But in a thermodynamic sense, all of these things are on the left. And this does not just apply to biopolymers. If you look at essentially, metabolites and cofactors, like ATP, all these phosphate bonds hydrolyze. The linkage between the sugar and the base will hydrolyze. Down here on the right in the middle, I have well, let's look at the NAD on the left. This has so many hydrolyzable bonds on the left. So, and then here in the middle, I have acetyl coenzyme A. This is a I don't know if you guys can see my mouse. That's a peptide bond that will hydrolyze. There's another peptide bond. These phosphodieesters will hydrolyze. The base sugar bond will hydrolyze in this phosphate bond. So what it means is if you put this molecule in water and you go away, it's gone. I mean, maybe it'll take a day, maybe it'll take a month, but it's thermodynamically unstable and it will fall apart into very, very small pieces. And this applies to everything in biology. Nothing in biology is stable in water. Maybe that's not true. Very few things in biology are stable in water. So there's two ways to think about it. None of the universal biopolymers in life are thermodynamically stable in water. You could say that the other way. None of these things will form spontaneously in water. Almost nothing in biology will spontaneously happen at high water activity. And the same applies to most metabolites and cofactors. And or say it in the forward sense, if you just put a cell in water and go away for a long enough time, you will come back, you will just have very small chemical species that will hydrolyze. All the polymers will go away. The phospholipids will hydrolyze. Everything will go. So what does this mean? This is what we think it means. It means life did not originate in water. We think it is virtually impossible. Nothing in biology is thermodynamically possible in water. We think life originated on land. And you might say, okay, so here's some land. There's no water here. What this means is that things are thermodynamically stable here. But we don't really imagine the land looked like this. What we really know is we have a spinning planet. And what the spinning planet does is it cycles water activity. So if you look over the surface of the Earth, the activity of water goes down in the day and up at night. So a spinning planet like ours has cycling water activity. If you ever, I don't know about Texas, I've never really looked in Texas, I don't know, but in many parts of the country, like I'm from Seattle, if you go out in the morning in Seattle, your windshield has water on it. By the afternoon, it's gone. And you come back next morning and it's back. So you have this cycling of water activity that causes water to condense and evaporate and condense and evaporate. So this cycling of water activity, we think, was really critical for the origin of life. But there's another problem with this desert here, which is that there's no organic chemistry on this. But what we think in the ancient Earth is that we really didn't have, well, sometimes we did maybe, but in general, if you look at what people are thinking going on in the ancient atmosphere, it was at periods of time, it was like kind of like Titan. Like Titan is covered with dunes of organic material. This happens in the atmosphere when you have a reductive atmosphere with certain molecules in it, you have dunes. And so, for example, Catling and Zonley think the cumulative organic production of, this is caused by the reductive potential of impacts, ranges up to a half a kilometer deep in organic gunk. So we should not think of the ancient Earth like this desert. We should think about it as really deep, deep organic gunk. And that organic gunk is very complicated mixture. In fact, we don't really, I've talked to Nick Wogan and some of these people. I said, I want to know everything there. Tell me all the species in that gunk. And they feel like, oh man, it's just so many we don't know. And I said, well, you say it's cumulative of a half a kilometer. I said, how deep is it? And what do you mean? What's the packing density? And they're like, oh, we'll get back to you on that. So I don't really know, I don't think the models are so good yet, but I think ultimately we will know a lot more about sort of the generation of organics. But the idea is this: imagine a rich, complicated, organic, sort of tarry, but with some small molecules, just getting wet and dry on a 24-hour cycle. This is the sort of conditions we think that spawned life on Earth, that cycling water activity in a rich organic environment. By the way, if anybody has any questions, you can totally feel free to interrupt me. Okay, so this has kind of caused a transformation over the last few years in how we think about the origin of life. We now, many people, including my lab and Nick Hudd's, I think this thing was really spawned, I think, by Ram Krishnamurthy and Nicholas Hudd. They did, I think, some of the very first experiments here, where you can take organic materials and you can just start to cycle the water activity, and you can do chemistry. You can make very, very interesting molecules. You can oligomerize things, and they just start to grow. And so, so the chemistry is very easy. We can make sort of proto-biological molecules very, very easily now by wet-dry cycling. And just, I think I have a chemistry picture. Yeah, focus on the left-hand side, and I'll just sort of show you, sorry, on the right-hand side. If you have, this is on the very left where my mouse is. Hopefully you can see. Oh, wait, let me try this. Oh, yeah, can you guys see that?
Yes.
Okay, good. Okay, that's a hydroxy acid. And then this is just a hydroxy group, right? You put those things together in water, nothing happens. But you dry it down and you make what's called an ester bond. And then if you have what's called an amino acid close by, you get what we call ester-amide exchange, where this nitrogen attacks that carbon and it switches it to a peptide. So all you got to do is put simple organic materials in solution, and you can create protobiological molecules. This is like, I would say over the last 5 or 7 years, this has been a total revolution. Before, people were taking amino acids and putting them together and trying to make peptide bonds, and it was just impossible. You could only do it by heating, but you had to get them so hot that it caused all kinds of bad chemistry of the side chains, So this all happens. This happens at body temperature. I mean, this happens at 37 degrees. You don't need, this. It's very simple chemistry, and you can think about this as kind of a chemical evolution in the sense that we form something with a very low activation energy. That's an ester. And then that is converted to something, an amide, which actually has a very high activation energy to hydrolyze. So it's a ratchet, right? You form something that's easy and you do it in 2 steps and then you can't get out. So you have this chemical ratchet that is just caused by cycling the water activity. I'm convinced that this is sort of a really, really important consideration for the origin of life. And what you get out of these things are peptides and some esters, and we call those, this thing at the bottom, there's a peptide bond here, and there's an ester bond there. This is called what we call a deoxypeptide. Those are the oligomers we get out of this so far. Okay, so trying to advance, I guess, because I have that pen on. There. All right, so now let's think of, I still have those. Okay, I don't know what to do about that. Okay. Let me think about that now. I'm leaving them there. Okay, so think about what this means for the origin of life. You need water, but you can't have too much water. And you need a spinning planet. A tidally locked planet, we think, will not do this. You got to cycle the water activity. The way we think about biology is really all about making things by condensation and breaking them by hydrolysis. And we think that is chemistry taken from the surface of the earth, right? The surface of the earth, the water activity is cycling. You put water in, you take water out, and that is the chemistry of biology. And we think biology just picked that right up from the surface of the earth, but that's not going to happen in a tidally locked planet. I will say, and you need land, right? If you have just an ocean planet, then you're doomed. Now let's think about some other ways you can cycle water activity. For example, you can cycle water activity with freeze-thaw. It doesn't have to be just evaporation and condensation. So you look at some of these moons of some of the large gassy planets, which covered with ice, and then you have this gravitational flexion. And I would imagine that you have freeze-thaw cycling, and which is also cycling of water activity. So there's a variety of ways you can cycle water activity. The sun or a sun is not the only way to do it, but it's the easiest way to do it. Maybe if okay, I guess I'm going to leave those circles there. Okay, so oh God, you know what? I'm gonna maybe if I stop sharing and come back.
It's under view options.
Is it under view options? View options. I'm looking at view options. I don't even see view options.
Yeah.
There's this thing down here. Oh no, that's well. Okay, you guys, just forgive me those circles. I don't want to. Okay, so we. I need to go backwards here. Yeah, we wrote this up and sort of talked about this in some journal, and then Mike Russell, who maybe some of you know, Mike is a friend of mine, but he was offended by these ideas, and he wrote a paper actually in which he kind of went after us very specifically, which is fine. I'm a big boy; he can do that anytime he wants. But what he said is that my model cannot be right. He says that the early Earth was a water world. It was all water. Nothing was less than 4 kilometers deep. There were no Icelands or Hawaiis. There was nothing but water. So I read this and I thought, okay, there's a couple possibilities, but the possibility I favor is that prebiotic chemists are going to have to tell geologists about the ancient Earth, because obviously life originated, and life, I believe, had to originate on land. And since we're here, this model cannot be correct. Mike Russell had to be wrong, is what I decided, even though I know no geology. I'm just based on biochemistry and what I'm thinking about prebiotic chemistry. I said, this cannot be true. So I called up a friend of mine, June Cornega, and I said, help me out here. What do we know? We need the geologists to tell us what they know about the ancient Earth. And June said, let me look at it and let me think. And he read Mike's paper. And Mike had actually cited June heavily in his paper. And June called me back and he said, no, this isn't right. And ultimately what he said, so in his review, and I have to say, I said, please write this up, do it. So I'm not like totally not involved in this, I have to say. But anyway, I didn't write June's paper. He doesn't need my help to figure out any of this. And he concluded that no, he thinks that there may, in fact, he thinks there could have been continents, but there certainly was isolated islands in the late Hadean and early Archean. So anyway, June's conclusion was that that the possibility of land is open and it's very likely. And then actually what happened was there was a long email exchange between me and June and Mike Russell, and we kind of all agreed that yes, that there is a possibility of land. So the idea that the ancient Earth was a water world is, I mean, we can't exclude it. I exclude it because I think if that was true, there would be no biochemistry. Biochemistry could not have happened on a water world. It's just not possible. I don't believe. So, okay, I'm about done here, but I want to conclude this. This is the problem, and this is what, like, why geologists, I have to say, I've talked to geologists in general about this. Even June Cornega, if you read his paper, he's like, I like thermal vents, I like the ocean, I want life to originate in the ocean. So this is the problem, is that it's not possible, okay? Nothing in biochemistry is possible in a thermal vent, okay? You cannot do condensation in a thermal vent. Now, I've talked to a lot of geologists and they will say things like, well, on these surfaces, this can happen or that can happen, and maybe you can do dehydration in some strange surface phenomena. But you know, no one has shown that. And we know you can do it very easily by dry down experiments on land. So in these weird surfaces, there's never been any demonstration of any condensation, dehydration reactions happening in anything related to a thermal vent, nor has anyone observed any kind of non-biological polymer coming out of a thermal vent. So I think that it just seems, you can't exclude it, but the whole thermal vent thing seems terribly improbable to me. Now, that doesn't mean there's not biology in thermal vents. Of course there is. There's lots of biology. There's incredible biology there. But the origin of life, I would say no. I think it's very, very improbable, because thermodynamically, essentially everything we know about biology is impossible in a thermal vent. So that's what I wanted to tell you. So this is my group, and I hesitate to circle anything, but Moran Frankel-Pinter worked with me on this metabolism project that was a lot of work, and Vahab back here we had to go through like thousands and thousands of biochemical reactions and count up all the water molecules and all that. That was a massive amount of work. So they helped us, and I would like to thank NASA for their support, NSF for their support, and Georgia Tech. So thank you guys, and I would like to talk, and I know that in general, I've talked to geologists before, and they just do love thermal vents as the origin of life. But I have to say, I think it's just, I'm sorry to say that it's just very improbable in my view. So.
Okay, we've got what, like 3 different or 4 different questions already. You want to read out
Yeah.
the first one?
Okay. First of all, I'll thank Loren for an extremely provocative and interesting talk. I know you sort of took many of the ideas that I've always had as a naive astronomer and turned them on my head. So I have to think a lot about what you said.
And Lucy's question.
Yeah. Okay. There's a couple of questions in the chat. Actually, I'm going to start with my own question for you. Since you say it's difficult, if not impossible, to start life in a purely aqueous environment, I know there's a lot of people here who are building an instrument that they're going to fly to Europa as part of a huge NASA search for life there. So is it possible with your current thinking that life could have started in the surface ocean within Europa, or did it need something else, or beneath the ice?
Yeah, well, freeze thaw can cause cycling water activity. So I would not exclude it. One thing we know that I think is important, like if you look at Titan, for example, you have this incredible inventory, this organic, it's snowing polymers on Titan, right? And I guess I don't know enough about Europa. I mean, atmospheric chemistry can just produce this, a reductive atmospheric chemistry can produce this incredible inventory of organic molecules. And that's, I think, an important consideration. I don't know enough about Europa. I mean, do you imagine there, I guess I don't know. Is there like a source of organics in these oceans? If there's no organics, then that would be difficult. But if there's organics there, and freeze thaw can cycle water activity, so
Okay,
especially these
I guess
gravitational
there are
fields.
organics in the ocean, and you should be getting freeze thaw at probably the water ice interface.
Yeah, and I think also because of the gravitational field, I think there's this sort of flexion of the system, and I think that can cause sort of freeze thaw, cycling freeze thaw. So yeah, it's possible.
Okay, I'll read some of the questions that were put in the chat here. Grant Gatchis is asking, how important is the deuterium to hydrogen ratio of the water for biochemistry?
I would say I don't think it's important. I mean, I think our life evolved in a certain ratio, and if you just changed it now, you'd kill us all. But I think that if life had originated in a different isotopic environment, then it would all be good. I don't think there's, that some of the reactions would be different. Maybe some of the molecules, it'd be sort of different chemical selection a little bit at the early phases, but the principle is fine. There's nothing. I do believe, if you actually just think about our biochemistry, what I think determined the molecules was the prebiotic inventory, the day-night cycle. I bet if it was a faster or slower day-night cycle, we would probably have selected for different kinds of molecules, because you're selecting on the basis of hydrolysis and condensation. So, I think all of this is contingent, including contingent on isotopes, because reactions are, of course, dependent on isotopes.
Okay, thanks. I have a question from Eric Hyatt. He says, it's my understanding that people believe you need agitation to create cell structures. Do you believe this is true? And if so, where does the agitation come from in this diurnal evaporation cycle?
No, I don't believe you. In fact, okay, this is another thing that I think, when we our minds are kind of bent by looking at biology. Biology, we have these wonderful partitions, membranes and compartments and all that stuff. But I think the way I imagine it on this organic gunk, I don't think you needed that. I think that the partitioning was effectively a function of viscosity. And if you look at the phylogeny, the fact that bacteria and archaea have different membrane components sort of says to me that membranes are late. They all have the same translation system. That goes back deep, deep, deep. The translation system seems to be much older than the biosynthesis of membranes. I mean, I think there's no escaping that membranes are kind of a recent addition, and I don't see why you need membranes, because in the world I'm imagining, you can achieve a biological partition with maple syrup, basically. I mean, viscosity will give you everything you need. You don't need membranes, I don't think. In early, I mean, you need membranes now, but in early biology, I don't think you needed membranes.
Okay. Yeah, so I'll ask one from the room. The last universal common ancestor argument is suggested, we need thermophilic life to be where things started. So if wet, dry, and thermophilic are two things that you need, then would you envision that, on land in a hot spring kind of environment that has a wet, dry cycle is better than, say, your desert analog that has a wet, dry cycle? Or how important is the median temperature of that wet, dry, as far as you understand?
Yeah, I mean, I would, one thing is the last universal common ancestor is very far from the origin of life.
Yeah.
The universal common ancestor is very mature, sophisticated, I mean, cellular life form. And when I'm talking about the origins of life, I'm talking about the earliest chemical evolution, like where did these molecules come from? And I'm not sure we can use phylogeny and to sort of get back to that. So I don't, it's sort of funny. We don't have any time stamps. We don't know if the beginning of chemical evolution to Luca was 10 years or 10 million years or 100 million years. We don't know that. So I think it's very hard for me to even think about that kind of question.
Okay, yes, another question from the room.
Yeah, I'm wondering if you think that there's any possible scenarios where this essential water activity cycle could occur on a tidally locked planet, like maybe involving eccentricity or.
Wait, I didn't quite hear. Could you just say that one more time? I didn't quite hear that.
Oh, should I just speak louder? Okay.
Microphone is over there.
Okay. So I'm wondering if there's potentially any scenario that could allow the water activity cycle necessary for the origin of life to occur on a tidally locked planet, involving maybe the planet's eccentricity or a binary situation where the planet's orbiting an M star that's orbiting a G star or something.
Oh, yeah. You know what? I sort of hadn't thought about that kind of thing, but yes. Yeah, all of those are going to give you thermodynamic cycles. And so what I think is that if you have organic gunk and you have a thermodynamic cycle like this, you are going to do chemical evolution. I don't think it's hard. It doesn't take reagent grade chemicals and mass spectrometers. I mean, it's just really easy and it goes. But depending on the conditions, you're going to get different answers. So if you had a really slow cycle, you're going to select for a different kind of molecule that hydrolyzes and condenses at a very slow rate, but you're still going to get something. I think it's unavoidable. So yeah, I would not exclude any of those things. That's right. That's
along those lines, we have to remember that it takes a while for a planet to become tidally locked. And so if during that time, it would have an eccentricity, it would not yet be tidally locked, you could develop life, and then the planet subsequently becomes tidally locked, which is the way we find it today. So,
Yes.
there's an
Even-
easy scenario into that.
I think even like imagine the Earth was tidally locked. We still have seasons, right? I mean, because isn't that true? I'm not an astronomer, but I'm assuming because is that right? Maybe
Yeah,
not.
as long as you have
Yeah.
no liquid. Mhm.
Yeah. Okay. So yeah. Okay. So I guess I want to back off a little on the tidally locked thing. I think there's other ways to cycle water activity there. Yeah. Thank you for that.
Yeah, okay, there's another question here in the chat from Brant, which is, does the salinity of water play a role in this?
Oh, absolutely. Not just the salinity, but the, we think that life originated in an anoxic environment under high iron. And that iron was very important. If you look at RNA in the presence of iron, as long as you keep the oxygen out, so there's no Fenton chemistry, is a much more interesting molecule than RNA in the modern world in the presence of magnesium. So we think that RNA was selected in a world of iron 2, okay? And so that's just one, so it's not just the salinity, but it's actually the specific kinds of iron that, the specific ions that were available. So yes, that's all of that. The way to think about it is what we have is just contingent on what was there. If we had different conditions there, we'd have a different answer. I think we would still have, molecules that condense and hydrolyze. We'd have some things, but we would, we'd have molecules made with different lifetimes that interacted with metals differentially. There's many possibilities. I would say, especially for nucleic acids. Protein is maybe inescapable for reasons that I can talk to you about. But nucleic acids, there's different sugars, there's different bases. I think there's a lot of different possibilities for RNA. I think those molecules especially, I think, are very, very contingent on the conditions, I would think.
Okay, and it looks like we have one last question here from Eric Hyatt. Can you create these molecules in chondrites prior to impact?
Yes, I mean, we're talking about the sort of organic. Okay, so first, let me clarify. I think there's two sort of phases. There is the kind of what we would call the building block phase or the inventory phase, and that would be either atmospheric chemistry or chondrites. And then the second phase is the dehydration condensation, the taking of those molecules and just selecting based on their water chemistry. And those are basically two very, very different chemistries. Like guanine, for example, or bases don't come from dehydration chemistry, but they are linked to sugars by dehydration chemistry. So you need to, and the sources of these building blocks, we call them, can be also, I mean, the natural world can produce these in a variety of ways.
Okay, are there any further questions, either from the room or from those online? Okay, well, let's all thank Loren again. And this is exactly the type of really interdisciplinary process that we really enjoy here. So, thanks so much for doing this, Loren.
Okay.
We appreciate it.
Thank you, guys. Thanks for listening to me. I appreciate it.